All right. So welcome everyone to our next Malaysian Research Insider session in 2021. I hope everyone is staying safe and healthy. To those who are new to the Research Insider session on MBIOS Facebook page, providing a chance to everyone to discover our talented Malaysian bioscience researchers and a platform to interact with them. Each scientist will discuss their work, their experience in science, and how they got to where they are now. This conversation will be accessible to all and are directed at non-specialists. The general format is that the speaker starts with an overview of themselves and their topic of conversation, followed by a Q&A session. If you are interested in science or learning about topics outside of your area of expertise, don't forget to tune in every third Saturday of the month to chat with our scientists and learn something new. So I am Janatul Rahman, a former master's student in science at University Malaya. So this science chat is organized by MBIOS and media partnered by 100 Scientists Malaysia and Science Media Center Malaysia. A quick reminder for the audience, please feel free to post any questions in the chat box at any point during the talk. So our speaker for tonight is Dr. Jue-Sheng. So Dr. Jue-Sheng is currently a Postdoctoral Research Officer at the Queensland Institute of Medical Research. His research focuses on using statistical models to understand how germline genetic variation contributes to human traits and disease. While he primarily studies cancer, he has also worked on other fields such as nutrition, psychiatric disorder, eye diseases, and other behavioral traits. So without further ado, let's welcome our speaker for tonight, Dr. Jue-Sheng. So over to you, Doctor.
Thank you for the very kind introduction. Sorry for the slight delay. And thanks again for MBIOS for inviting me to share a bit about, I guess, my journey going from not a very science-heavy field, which I'll go through later, and how I end up doing biology, which is a shock to both my family and my friends. So I'm Jue-Sheng, as the host have introduced me. People can label us as genetic epidemiologists, computational biologists, or statistical geneticists. It's kind of the same thing. So what we are interested here is that we want to understand how genetic information can help us understand about disease trait variation, like phenotypic variations. It can be about a specific disease. It can be about a specific risk factor, and how the belief is that as we map more genes for a specific disease, we can unravel the etiology behind complex traits. So this had been my focus since I've started my PhD, which is very heavy on causal inferences, but I've also worked a lot on things like conventional GWAS publication, which is basically large-scale genetic studies to try and map causal or putatively causal genes that are related to a specific trait of interest, or it can be cancer, it can be neuropsychiatric disorders, it can be coffee intake, you name it. And I've been very lucky to have been working in this very fascinating lab with very nice colleagues. Everyone is really friendly, and because of all of this positive reinforcement, I have quite a remarkable publication record, which I'm really thankful to my mentors and my supervisors. And I actively collaborate with a few large-scale cancer consortiums like the Ovarian Cancer Consortium, the GenomeL, which is for melanoma. And I recently, actually not so recently, I graduated from my PhD in 2019, and since then I've been working in the same lab for the past 2 years. So my academic journey is slightly unconventional, to say the least, given that I have close to no biological background since from high school. In high school, I took at math. I was in the sub-science stream, so I swapped out biology for accounting, which I didn't do very well as well, and I did physics and chemistry. And then during my pre-U, I did it in Sunway University College. Back then it's Sunway University College under the Murphy program, which is Monash University Foundation. And I took, again, sort of the hard science subjects, swapping out biology for accounting. And I think I did computer science instead of chemistry. So I kind of have I was sort of prepared to go on, things like engineering or computer science. And then I did quite well for, I think, mathematics. And I got an offer from ANU to do actuarial studies because I think back in 2010, actuarial studies was one of those, top career choices. You are like the top 10 best earning jobs in Malaysia or top 10 best earning jobs in US. So I went on the hype train and tried to become an actuary. So I did a Bachelor of Actuarial Studies in ANU, and I would say this is one of the first transition that I got from trying to see that actuarial studies is more than just applied math. There's a lot of statistics to it. There's not just insurance math. There's a lot of beauty of statistics that can be manifested in different ways or in different fields that you might not just see, the financial aspect of how statistics is being used. So during my undergraduate times, I didn't do a lot of commerce-heavy electives. Instead, I chose to do mathematics. So in fact, all of my electives, I chose to do things like view analysis and third-year probability course, which opened up some gates, opened up some doors. And when I did my master, I told my parents, I kind of didn't like to do finance. I like the math aspect of it, but I'm not very good with written exams. I didn't find a lot of joy with, calculating interest rates and back then you have things like, runoff models in actuarial science. I mean, these are quite fancy mathematics stuff. It's fun stuff, but it just doesn't click with me. So when I did my master's, I was kind of in the lowest point of my life. I was like, okay, I have an actuarial degree, but I don't want to be an actuary. I don't want to go into finance. I don't want to go into commerce. What can I do with it? Then I came across one of my master supervisor, which is an astrophysicist. He's an astrophysicist in the Department of Mathematics, studying bioinformatics. So I was like, aha, this mentor is going to understand where I'm coming from. So I'm quite intrigued by how genetics can be applied. So this is about somewhere at this point of my academic journey, how genetics can be applied in a mathematical framework, or rather the other way, how mathematics can be applied in the genetic framework, and how you can use things like Markov chain, which I've learned in my undergraduate times, to try and work out transition probabilities, can be used in the same way to study mutations, to study nucleotide polymorphisms, and you can do phylogenetics. You can do a lot of fancy stuff with the same type of statistics that I previously learned during my actuarial studies program. So I start seeing mathematics in a very broad way, that it's more like a tool set for us to pursue whatever we want to study or we want to investigate, rather than a field by itself. So because I liked the bioinformatics aspect of it, about big data, there's a machine learning, there are a lot of different statistical approach, then I took a master's program in mathematical science, focusing on mix and match of different type of mathematical projects in quantitative genetics. And when I graduated from my master's, I started looking for PhD programs everywhere. I went for, I think, one of the bioinformatics conference in Adelaide, and then I met a few researchers from QIMR, which is not my supervisor, by the way, and I I just tried. I sent my CV to 10 different or 20 different research labs, and eventually my supervisor Stuart met me when I was in I think it was MASCA conference. So it was like a Malaysian sport event for all of the Malaysian student bodies, and I was there, and my supervisor was like, hey, do you want to do a PhD since you have a pretty robust statistical background? And I said, yeah. I started my PhD in 2015, working on something totally different. It's not on quantitative genetics, it's not on bioinformatics, it's purely on genetic epidemiology, which I'll talk about a bit later. And since then, I've graduated in 2019. I've been a postdoc research officer, publishing a lot of fun stuff. So why did I guess, as I described in my own life story, why did I step into academia? I find academia is one of those fields that you can have a lot of flexibility. Sorry. So you can have a lot of flexibility. You set your own time. You don't work 9 to 5. You can work 9 to 5, but you have your own time. You have your own target. You are basically judged by productivity rather than a specific schedule. There's a lot of independence in academia as well. You can try new ideas. If ideas fail, that's fine. You just try something new. And there's always a lot of room for you to express yourself or express a new angle into thinking about a specific research problem. And definitely because it's competitive, it's fun. You don't always win, but when you win, it's worth it. Publications, they are rewarding. The recognition from your work by your peers, these are all kind of positive motivation into kind of supplementing the fact that academia might not be everyone's top choice when it comes to finding a job. So obviously, because I come from a totally mathematical background, some of mathematics, statistics, commerce, going straight into a PhD in genetics is a bit of a challenge. And I believe a lot of the audience here, if they're in science or if they are working in the same field, they will have this kind of feeling like an imposter syndrome. Throughout my PhD, I have times where I always felt like I'm an imposter. But the more you think about it, it's actually an opportunity to showcase things that that others didn't know, that you might actually have an upper edge. So I would like to see it this way, that everyone has imposter syndrome, and we just have to work out. We have to work out with that level of insecurity, and eventually try to see the positive side of having a multidisciplinary background. And so this is a bit from my own experience. When I did my master's program, I've always been kind of like not really the black sheep, but the odd one out. So when I did my first master's project, I was in a lab that was focused on studying ecology of honeybees, like the interaction of honeybees, a little bit of genetics work. However, I was being recruited to work on Proritis, which is a species of plankton, I think. Yeah, it's a type of symbiont in the sea, like coral and symbiont. So I was being tasked to develop some kind of mathematical framework that can help separate transcriptomes that belongs to a host and transcriptomes that belongs to a symbiont in a mixed sample of in a mixed biological sample. So and then the next thing that I worked on was on amino acid transporters. So I was working in I think it was called Stefan Boer in ANU, which is a very famous biochemist. So his lab is mainly interested with understanding how amino acid transport, so amino acid uptake, can influence cancer cell growth, and whether you can isolate a specific type of protein. Most of, I think 80 or 90% of his students have PhD projects or some level of research project done on mice knockout studies. But what I've been tasked to do was a bit different, was that I was paid to write formulas, mathematical formulas that is based on enzyme kinetics mathematics to try and predict what's the cell homeostasis based on a specific parameter. So slightly sort of the same research field, but a totally different angle. And lastly, when I went for an interview for my PhD project, you're expected to know, you're expected to be someone from epidemiology, public health, statistics, know a little bit about cancers or disease. Not quite the case for me. I have a semi-commerce degree, and I have an actuarial degree, so I have my almost 80% or 90% that I've known about statistics have been applied in finance, applied in interest rates, in claim distributions, and I'm working with, in my master's, I did stuff on population genetics, which is not quite the same area of genetic. So obviously everyone will have this kind of an experience that sometimes, you might not feel like you're the best candidate for the job, but eventually with enough persistence, you can, you will go through, trust me. And this is just a summary of the stochastic nature of what we think we work with and what we end up working with. So the plot on the left is the advertised research focus for my lab when I started my PhD. And on the right is retrospectively what I've worked on during my PhD. And obviously there are stuff that are solely from like candidates' input. So for example, the pan-cancer analysis is one of the angles that I've proposed to the lab that we should start studying. It's a very interesting angle to look at different types of cancers and draw large-scale causal inferences. So my daily life as a researcher, as an obligatory slide that everyone has, is nothing too different from everyone else. There's always a difference between expected and what actually happened. But I'm just going to show you another dynamic to it, which is the pre-COVID and the during COVID angle. So pre-COVID, a lot of my time is spent on writing manuscripts, studying papers, coding, analysis, designing studies. During COVID, kind of, because of the work from home arrangement, you can kind of slack a little bit. You can kind of mix and match your own timing. You draw your own targets. So sometimes in the morning I'll walk the dog. I'll spend the whole afternoon procrastinating, and I'll do grocery shopping on a weekday because everyone does it on the weekend during the COVID period. And then, you schedule your Zoom meeting around kind of these three priorities sometimes. And then, sometimes after dinner, I start doing some writing, analysis, or discussion, and work on weekends. So there's not really a 9 to 5 rule that neither I follow nor did my lab heavily enforce. So there's a lot of flexibility working with the research team that I have right now, which is kind of great. And actually, when I talk about writing, analysis, and discussion, you will like to think that, oh, every day is going to be as productive. That's not the case. Sometimes, you end up Googling very trivial things like, how to write basic if statement in Bash. Sometimes you have to chase up conflict of interest form out of 48 authors. If you are dealing with large-scale consortium, that's not a very good use of your time. Cleaning spam emails, deciding which one are legit, which one are just predatory journals. Because of my background that's very quantitative, I sometimes have to go into WebMD to try and understand a bit more about the biology behind each cancer, so that you don't write introduction to papers that are embarrassingly wrong. This is where I spend most of my time fixing very trivial bugs in your scripts. It can take a day, sometimes it doesn't work, you will just do it the next day. And then, yeah, it's a lot of time spent on kind of cosmetics as well, which is basically trying to make your paper look aesthetically beautiful, so that you can push it into some more fancier journals. So this is the reality of the life of a quantitative researcher. And if I would be able to break them down into guesstimates of average time spent per week, again, because it's average time spent per week, there's a lot of fluctuation from week to week, because sometimes you can be very productive, sometimes you just don't feel like working at all. This is a very nice breakdown of how I spent an average week, although that's no longer the case during COVID. But I try to maintain that balance. And just to point out that if you look at the research task that we have, it's a big challenge, that because for every successful researcher, you need to be able to balance between academic skill, people skill when you're negotiating project leads in large collaboration, or discuss cross-field collaboration with oncologists, with clinicians, with psychiatrists. And there's a lot of management skill, because you want to avoid overcommitting for a small favor than getting your main projects done. So how does it really feel like to be a gene mapper? At this point, a gene mapper is basically people like us that we try and map genes for disease, and to try and understand what actually causes this disease. So a gene mapper is basically sort of a geneticist to an epidemiologist, because epidemiologists traditionally work on traditional risk factor. They don't really understand much about genetic risk factors. But at the same time, you have to be a statistician in front of molecular biologists, because they know more about biological pathway, functional genomics, pathological variants, and all the whiz words in functional genomics. So when you are facing a molecular biologist, you act more like a statistician. And then when you are talking to a clinician, you have to talk like a data scientist to tell them what all this big data means. And the funny thing is that during TV interviews, because there's a perception that every scientist wear a lab coat, you'll sometimes be forced to wear a lab coat. So yes, the gene mapper is basically someone that have to be very prepared to wear different hats in different scenarios. So what my research is all about is basically can be broken down into 3 different areas: the gene mapping to identify variants related to the specific disease. There's some functional genomics follow-up to try and understand what this specific mutation translate to. Is it on a coding gene, or is it on a regulatory region? And what does it tell? What does this tell us about the pathogenesis genesis of the disease? I focus more on running causal inference analysis, maybe because of my background that's more heavier in the statistics end, which is basically to try and understand how genetic variation is attributable to disease by that is consistent through either upstream or downstream influence of other risk factor. So the next question is, why are we interested to study common genetic variation? So the relevance is that it's quite relevant towards understanding how, for the past 20 years, there's a paradigm shift on how we study context trait genetics. So the paradigm shift, in the past, everyone will know about how we look at genetic and how it causes disease. Is that if you have an abnormal gene due to some mutation or inherited by your parents, then you can kind of work out the penetrance of a specific disease based on whether the kid is a carrier or is an affected individual. In this case, this is an example of what we call a recessive genetic disorder. in reality, the number of diseases that fit in this category is way lesser compared to what I would think of as a complex disease, which is basically disease with a complex mix of etiology that is not attributable to a specific single mutation. So this is a plot that I like because it basically tried to conceptualize what it means to have high genetic liability for specific disease. So every single gene will have a very small, sorry. I'll answer the questions later. Sorry. So what this means is you can have some kind of a genetic liability model where if you aggregate enough genetic liability through these different small size mutations, then you can it can then manifest as a disease. So you can conceptualize it as, these are the unaffected individuals are basically, like the controls in a standard case control breast cancer analysis, and the affected individuals are the individuals that are actually diagnosed with breast cancer. And this is kind of the genetic liability that if you surpass a specific threshold, you become a case. So if you combine these two paradigms, you then get a very nice diminishing plot that basically disease that have very high genetic loading are those that are, these are very likely to be monogenic disease or genetically determined. And as you go down, you have a fair mix of both indirect genetic effect, direct genetic effect, and environmental effect. That's what we label as polygenic traits. So the question here is, why are we interested to study genetically inferred causality? So the core idea of genetically inferred causality is that how you can just rely on basic statistics. You don't need any biological insight at all, and just use genetic association to try and understand whether germline variation on a specific trait on the other trait is reflective of an underlying causal mechanism, assuming there's no external factors. So the ultimate task is to estimate the relationship between a risk factor of interest and an outcome of interest, relying on just genetic association. And this is also a framework known as a Mendelian randomization for those that are more familiar with instrumental variable analysis. So in a typical GWAS, you can have an association between a specific genetic marker and cancer, and you can also have a association between a specific genetic marker and, let's say, 25-OHD is basically vitamin D. So to draw a causal inference between vitamin D and cancer that is unbiased from environmental confounding, what you can do is you can use a strong genetic marker that is strongly associated with vitamin D and assess whether this variant is also associated with cancer, because if that's true, that tells you vitamin D is likely causal on cancer based on a very simple statistical framework of instrumental variable analysis. So how is this type of genetic causality inferred? Basically, you have some randomization analysis because genetic variants are randomized at birth in a population. So if you think about just one specific variant that's associated to vitamin D, if you have the AA genotype, for every A allele that is vitamin D increasing, you can think of it as a lifelong predisposition to have higher levels of vitamin D compared to someone that doesn't have the A allele. So obviously, individuals that have the CC genotype will be the baseline, will have the baseline vitamin D level. And obviously, the trait of interest that we measure, let's say for like cancer, is much later in life. So what we are actually capturing here is a lifelong predisposition of vitamin D that is predicted by a gene. These are just some very basic formulation of how it works. And this is to draw a parallel on how Mendelian randomization framework that is basically built from genetic causality. It's different from traditional randomized clinical trial, where if you have the risk allele, it's equivalent to you being put into the intervention group. And then if you don't have the vitamin D increasing allele, you are in the control group or the placebo group, and you can then compare the outcome between these two groups. So this is one very interesting toy example that I like, which we tried, and it wasn't actually part of my PhD. It was just I got some inspiration out of this project from talking to some colleagues and that's working on taste genetics. So we tried to use 3 different genetic instruments that can predict, that are shown to be associated with 3 different beta taste perception. So this is the prop, we call it the prop SNP. So if you have a variant, the mutation in this, if you have the mutation in this variant, then you are on average more likely to hate the taste of Brussels sprout, because you can taste the intensity of the bitterness in Brussels sprout. And surprisingly, bitter taste perception is linked with how likely we drink coffee, or how much we like coffee, or what's the relative risk that we end up becoming a heavy coffee drinker. And as you can see here, if you have heightened sensitivity on prop, you end up drinking less. You end up drinking less coffee but more tea. And this is very interesting because I actually took inspiration from this work from my colleague and dear friend Daniel, which is a taste geneticist. And I just sent a very friendly email to Marilyn Cornelis, which is one of the leading author that led the largest coffee genetic study. And we collaborated and wrote this paper, which basically showed how bitter taste perception is causally linked with how much coffee, tea, or alcohol that we drink. And surprisingly, this is listed as the top 100 papers in Scientific Report in 2018. So that's a really good outcome. And so moving from a single SNP, if you want to deal with traits that doesn't have straightforward etiology, like like height, where we have 2,000 different genetic loci that have been discovered to date. We can still use similar type of causal inference mechanism and look at overall risk on cancer, as we've shown here. You can actually collapse all of these different cancers together and ask yourself, okay, if height is causal on colorectal cancer, it's causal on melanoma, what about just overall cancer risk? If I'm not interested with any specific cancer, can we draw that kind of inference? And the answer is yes. So we applied similar statistical framework to study vitamin D, coffee, fatty acid, obesity, and these are all in some really fascinating journals. So I would like to show you a little example of how you can take a very simple idea, causal inference idea, and work on more complicated scenarios where to draw precise causal inferences. So this is to share a bit of work in progress that I did on balding and skin cancers. So the underlying idea is really simple. We know that observationally there is some people that are bald have higher risk of developing these 3 different types of skin cancer: melanoma, non-melanoma skin cancers. And we want to understand why that's the case. So we looked into genetics. Obviously, if you just pick genetic variants that are related to male pattern baldness, you can straight away do a simple analysis with this, because you have you can use large GWAS data on skin cancer, you have large GWAS data on male baldness. That's all you need. But what about some more complicated scenario? For example, we know that testosterone is linked with the risk of male pattern baldness, and there is some existing hypothesis that testosterone can be related to skin cancer through some androgenic pathway that trigger melanocytes. Or the other case is that skin cancer is also related to human skin tones, that if you have a very light pigmentation, you have a higher risk of skin cancer. Or another pattern of confounding, that immune system can bias the relationship between male pattern baldness and skin cancer, because if you have some compromise, you have genes that compromise certain immune response pathway, you actually loses hair, and that also have an independent effect on skin cancer. So as you can see, the triangulating causal inference is not always very straightforward, and sometimes it relies on more sophisticated mathematical frameworks like this, that you have to use multivariable framework to estimate the marginal effect for all these three different pathways independently. So that's just a flavor of how you can take some very simple mathematical framework and expand it, and to reflect the more realistic realization of how balding is related to skin cancer. So the problem with this kind of genetic inference is that, what about you have problems of trait heterogeneity? For example, there are hundreds of different ways things can go wrong. It can be phenotype heterogeneity. Whatever sample that you are using can be vulnerable to sampling bias. So it's no longer just a genetic problem, and we want to ask ourselves: are there systematic differences in trait variation in the context of disease? Is there subgroup difference in cancers? And I would like to emphasize this make it go away test, which is a very useful test that every scientist should do. Is that if you see a positive association, make it go away. Have you tried different ways to make it go away? That helps improve the robustness of any claim that you're drawing downstream. So this is one more complex example that I would like to show that I think encapsulates the type of work that I do really well. So we are interested with gastroesophageal reflux disease, or we call GERD, or some people call it acid reflux. It's basically a scenario where you have regurgitation of acid reflux up to your esophagus. It can happen after eating, or it can happen in a very chronic way. And the risk of acid reflux is that it's currently known to be the strongest risk factor for Barrett esophagus. And Barrett esophagus is really deadly because it's a precancerous condition that starts changing the distal esophageal epithelium and start changing it basically into stomach cell to counteract the increased acid exposure. And it leads to esophageal adenocarcinoma or esophageal cancer, which has really, really poor survival rate. So to understand esophageal adenocarcinoma is not so easy, because it's not easy to get people to participate in large GWAS studies, because EA kills you so quickly that we usually don't have a lot of case accrual. So this is why we study GERD. And in the past, there had been some study speculating how GERD is related to major depression. And this is again like a Mendelian randomization framework where they show there's some kind of bidirectional relationship, that if you are genetically predisposed to have acid reflux, you are more likely to be depressed. The same way, if you are genetically predisposed to be depressed, you are also more likely to experience acid reflux symptoms. So we have done some gene mapping work on reflux, and we have mapped 25 genes from I think 6 that's in 2016. So there's a major advancement on how much we understand about reflux. And this is just a very quick plot about the genetic correlation. So if we take the genetic architecture of reflux and we correlate it with the genetic architecture of these other traits, you can basically see that yes, there is a very strong genetic correlation between depression and reflux, the same way with BMI, which is expected since BMI is a risk factor for reflux and a negative association with education attainment. Then the major challenge with GERD, however, is that this disease or this disorder is heavily reliant on self-reporting. So if you look at how doctors diagnose GERD, is based on a specific list of criteria, like, you know, do you have bloating, or you know, do you have symptoms of heartburn? And there's a lot of all these symptoms that later there's some clinical studies that found that if you do actual endoscopy, 40% of people that report having GERD or reporting having these GERD symptoms don't really have abnormal, only 40% of them have abnormal pH exposure. So the remaining 60% might fall into what we call esophageal hypersensitivity, or what we call functional esophageal disorder, which is related to this gut-brain axis, that you have heightened pain sensitivity, you feel nausea, you feel uncomfortable, you have bloating, you have problem with bloating, but you don't really have abnormal acid exposure along the esophagus. So there comes another problem. However, before we go there, we have to first understand, can we then use the fact that acid reflux have a multifactorial basis to our advantage to try and understand more about genes that are related to acid reflux? The answer is yes, by using what a statistical framework called multi-trait genetic analysis. So the overall idea is really simple, basically saying that if you have a lot of different correlated phenotypes that are correlated with your disease of interest, you can combine them in some kind of a linear mixed model framework and enhance your sample size by increasing power. So this is the toy example from the original paper that's published in Nature Genetics. It's not our work. It's from a group in the Broad Institute, which they basically show that, you know, if you combine subjective well-being with depression or with education attainment, you can generate a larger GWAS for subjective well-being. And as you can see here, measured by the mean chi-square, which is a measure of effective increase in sample size. So the take-home message is that if you lump up a lot of correlated traits together in a meaningful way, you can in fact increase power for your specific trait of interest. And so we tried that with acid reflux by combining BMI, major depression, and education attainment, using the largest GWAS we can find from the public domain. And then we also did one for Barrett esophagus, since acid reflux is a very strong indicator for Barrett esophagus downstream. So what we then see is that, yes, by doing this kind of multi-trait analysis, we almost double our sample size, and we went from finding only 25 genes to actually finding 88 genes that, although I didn't show here, they're actually replicated in the 23andMe cohort, which is really, really big. So it does help us enhance our ability to understand more about GERD etiology. And then the next part is to revisit our problem on clinical heterogeneity in GERD, because now we know at least a subset of people reporting acid reflux might not actually have acid reflux. So we attempt to try and understand why that's the case. And we developed some mathematical framework to try and split these results, all these 88 genetic loci back up, to try and work out how many is driven by BMI, how many is driven by MDD. And we do some functional follow-up to try and see whether these two groups, between BMI and MDD, the genes that they are associated with have gene expression differences across key esophageal tissues, and whether they show different genetic association with esophageal cancer. So we assessed first prediction accuracy, which is kind of promising, showing that if you would have naively just used GERD as a phenotype, so if you had just used the 88 genetic marker and regress it against esophageal cancer, you will see a positive relationship, which is what we already know. But once you stratify across those that are more likely to be confounded or linked by MDD, and those that are consistent with actual acid reflux that's driven by obesity, you then see, you start seeing some difference in terms of their effect size, where almost all of the association of the overall estimate is being explained by the obesity-driven GERD. So in parallel to that, we also look at gene transcription differences, to look at differential gene expression. And as you can see here, the obesity genes are way more pleiotropic, but what's more interesting is that for esophageal junction, esophagus mucosa, which is the three key esophageal tissues, you see differential gene expression for the obesity-driven GERD, but not the neuropsychiatric or the depression-driven group. So this is an example of how you can move from gene mapping into using different type of statistical mechanism to deal with heterogeneity in disease phenotype and then refine causal inferences. So obviously today what I show is just a subset of the active research area that I work with. So this is a list of some, half of it I think are collaborations, like the brain imaging work, Parkinson's disease and cross-ancestry work are collaborations, and then my main interest is still on pan-cancer analysis, a bit of eye disease and nutritional phenotypes. So the added bonus for working in science is that you not only get journal publications that make your work accessible to other scientists, you also get a lot of media attention in the public domain. For example, these are some of the news articles that they've written on our coffee and cancer work. More interestingly is the unexpected surprise from the bitter taste work that got extensive media coverage that I lost track how many there are. As you can see here, the online attention for that paper is ridiculous, given that this is actually not a major part of what I work on daily. So these are just perhaps some closing remarks is that it's always good to try and explore different avenues to try and transform existing research idea, implement new concepts by that you learn from different disciplines, and identify budding areas of research. And for those that are in their PhD, I would urge you to try and think about collaborations. Always try to work out if you think you can collaborate or you can contribute. Always feel free to offer your service. You never know. Sometimes, you might just be involved in got an opportunity to be involved in something really big that came out of surprise. Obviously, for those that are still writing their thesis, science is an ongoing process. Obviously, what I've shown you here is things that have worked. Out of 100 things that I've worked on, probably 30% made it out. So you don't expect everything to go as expected. There's no perfect thesis. If you have enough, you write them up, move on, work on the next problem. Don't be overly perfectionist about it. And finally, I want to acknowledge my research group. This is me here. That's my boss, Stuart McGregor, and that's Priya, which is my friend, mentor, colleague right now. And this is all, they are the members of statistical genetics, and my wife here for a lot of more support, and Cute Little Yogurt, which is our first puppy, and this is the younger sister. And happy to take any questions. Sorry if it went a bit overboard.
All right, so thank you so much for a very informative presentation, Doctor. So we have collected a few questions here. So the first question is from Zing. Since you are jumping across field, does it mean that you do a lot of self-learning? Because a supervisor from a biological field might not know what is happening in a mathematical model.
Very good question. So from my master's, the difference is a bit smaller because I went into bioinformatics, and my boss is still kind of, yeah, they work on bioinformatics. They might still have very similar bioinformatics background. It's just that because they are enlisted in the research school of biology, they are more likely to get biology students that work on more biologically oriented problems, that they are like lab focused, wet lab focused, and that's fine. For my PhD, I don't really have that, maybe a little bit, because my supervisor himself is from animal breeding, so my supervisor himself has a very quantitative background. So my supervisor himself has a very quantitative background, but but because the people that we work with, it ranges from epidemiologists, clinicians, oncologists, especially if you're working in large consortiums, right? You have people that are very biology focused, you have people that are very statistical focused. So yes, definitely you have to do a lot of self-learning, but it's not, I guess it's self-learning, but it's not isolated learning. So you can still share, you can still learn with different students. You have more biology-oriented students. You can ask them about very simple concepts. But definitely, if I were to think back, I think even if I'm a very quantitative background, if I'm from a very quantitative background, I think I spent a lot of my first 6 months in my PhD doing a lot of self-studying, because even though it's mathematics, mathematics is so wide that I deal with the very pure math type of mathematics, whereas the research that I'm tasked to do are very applied, and they are very very applied statistics based. So I end up learning a lot about hypothesis testing. I have to do a lot of catch up as well. Obviously, if you have a very good math background, it's a bonus, but yeah, you still have to look around and keep up with the level of self-study.
Yeah, that's true. All right, so for the second question is, what is your plan after your postdoctoral training? Any plan to return to Malaysia to pursue your academic career?
That's a very good question. I guess I've visited, I think, Cancer Research Malaysia. Shout out to whoever is from Cancer Research Malaysia. We should catch up soon. I visited Cancer Research Malaysia in 2017, I think, to try and understand a little bit about how things are progressing back in Malaysia. From my understanding is that although the paradigm shift had become very obvious in Western countries, but our understanding of genetics and our understanding of what type of people are tasked to do genetic research is still in the pre-1990 era. So we still think that if you have a biology background, you need a biology background to do genetics, but we are seeing more and more of the case that that's not really what's happening. And I talked to people from Cancer Research Malaysia in one of my academic visit there, and I think the stuff they do is quite fascinating as well. Obviously, there's still a lot of differences in terms of career opportunity and outreach, but because one of my academic goal in the long term is to focus on Southeast Asian genetics. So I've always had this belief that Southeast Asian genetics is unique by itself, because it's one of the growing up in a multicultural and multiracial society. I think Malaysia has a lot of potential in terms of genetic research, because you have you have a natural community that can help understand problem about admixtures, understand about different genetic risk prediction to different ethnicity in the same community. So there's a lot of I guess potential for Southeast Asian genetics to bloom. The same way how 2 or 3 years ago we have started seeing a lot of Asian biobanks emerging. So I think there is a lot of future prospect for Malaysia and Singapore. And the attitude I have right now is that I want to make sure I learn enough, or I'm able to get the most out of my time here, so that when I actually transition back to working on the Southeast Asian population, I'm someone that can lead some promising initiative.
All right. So next question, you publish quite a number of articles in a prestigious journal. Any tips to ensure the quality of your work or output?
So can you repeat that one? Sorry, I was reading some questions.
Okay. You published quite a number of articles in prestigious journal. Any tips to ensure the quality of your work or output?
I guess, all right, so that's a very interesting one that I wanted to put a few slides to share, but it's good that someone raised that up. There's a difference between being overly productive and being selective, and having a very good mixture of both. So I would maybe share a very small experience that I had. So my supervisor was a student of Peter Visscher, which is one of the key figure in quantitative genetics. So my supervisor is more open to different journal quality. So as long as we are not publishing in predatory journals, as long as it's some tier 1 journals, usually we don't spend a lot of time trying to finesse such that it goes into one of those, like Nature Genetics or Nature Communication. That's not necessarily the case if your supervisor have like an idol burden. Like if you're working with the best person, the best person in the field of quantitative genetics, sometimes you have the pressure that you need to publish in all these very prestigious journals, because your supervisor simply wouldn't let you just shove it to one of those lower tier journals. So I'm kind of lucky that I didn't have that kind of problem. So if you think about strategies, that we try to sell our work as high as possible, but we don't hold it off if it ends up going to something like human molecular genetics, which is still a fascinating genetics journal. And sometimes you'll be surprised, like for example the bitter taste and coffee work. We actually intended to send it to scientific reports anyway, because we thought, it's coffee, it's not cancer, it's not cardiovascular disease. The big journals are not going to be interested with it, so we thought of we just send a scientific report. And I kid you not, current even up to today, that's the alphametric score for that scientific report paper is probably the highest out of all of my cumulative work on cancer genetics, which is sometimes you can't expect or you can't predict this kind of stuff, right? So yeah, the tip is to basically just make sure if you're doing robust science, try to sell it. Don't oversell it. Sell it to a correct target journal, write it to the correct audience, and if you can get it in, that's good. If you can't, don't try too hard, move on.
Very good advice. All right, so next question is, in your opinion, what is the next frontier for genetic research?
So Mendelian randomization. So actually, what I've shown here is considered. It's sort of not really a frontier, but had been how gene mapping have progressed. So in the past, what we're interested with is that we are interested to map a lot of genes for a specific disease and try and understand what each of these genes does. And in today's world, because there's a lot of very large-scale genetic data out there, that you start doing a lot of digging or a lot of data mining. So in fact, a lot of the recent progress in drug repurpositioning So if you repurpose specific drugs, a lot of them have used genetic data. So basically say that, okay, if I found for, let's say specifically Huntington disease, if I find this gene is associated with it, can I then find what other genes can, what other disease is also targeted by this gene? And you can kind of complete this kind of very large association map to try and work out whether you can find a specific gene that is druggable and you can fix multiple diseases at the same time, or you can do a lot of like Mendelian randomization framework that you can find genetic predictors for a specific drug response, and then you can use that to try and understand drug resistance on a specific drug on, let's say, COVID or cardiovascular disease. So there's with the rise of big data, I think there's a lot of new omics data that is coming up. But do I then think that genetic analysis will become obsolete? I don't think so. I think we are still far from totally unraveling the entire genetic architecture for a lot of diseases. And you also have like new disease that come from time to time, like COVID susceptibility had become a really hot thing for the past 2 years, and you will always have new challenges that break the barrier between what's genetic and what's environmental. And the surprising answer to that is that a lot of things that we thought was environmental, in fact, have some level of genetic background. So I think it's we will probably still do very similar stuff in the next 10 years, but focusing on maybe different population like African population, Asian population, to try and start seeing start giving genetic studies a more global representation. I think you had yourself muted.
Sorry. Okay, we have a question from Ramesh Arif. Is there any scientific evidence that coffee may be related to a higher risk of cancer? So I'm a coffee lover, so I hope that the benefit.
Don't worry. Coffee probably doesn't do anything other than changing caffeine metabolism. I think it's been shown many times that coffee is unlikely to be related to cancer in any way. So yeah, it's not related to related to any increase of risk, nor does it protect you. So take it in moderation, I guess.
So I guess we have come to the end of the Q&A session. So thank you for the very informative presentation, Dr. Jue-Sheng. So, and that brings us to the end.
Yeah.
Thank you so much for all your interest and attention, all from the Zoom and Facebook Live. For those who joined late or missed our science chat, you can watch the edited video on our MBIOS YouTube page after this. So we will see you all on June 19th for our next talk of the year. So thank you, everyone.